Well, thank you. It is my great pleasure for today. We have Dr. Jing, he's a professor in Beijing University, Beijing, China, who will present multicolor magnetic particle imaging today in this webinar organized by nexMPI Cost Action, which is still open for people to join all over the world. Fantastic opportunity to work together on MPI imaging as a next best choice for medical imaging altogether. Well, Dr. Jing got his bachelor and PhD degree from Zhong University Science and Technology in China, and I met him as he was spending quite a few years under his Humboldt Fellowship in German Research Foundation as a PI in Braunschweig. So at some point he returned to China and basically doing wonderful work on MPI there as well. Well, without further ado, Jing, please, floor is yours. Let us hear about multicolor MPI from your side. Go ahead.
Okay, so thank you very much, Leila, for the kind introduction. So, okay, I don't know, good afternoon, good evening, and maybe also good morning, everyone. So it's my great pleasure to give a talk about multicolor MPI. So as all of you can see that, so the title is also very colorful. So I especially did it. So I'm Jing Zhong from Beihang University, so Beijing, China. So at the beginning, so I will give a short kind of self-introduction. So from 2005 to 2014, so I did my bachelor, master, and PhD at Huazhong University of Science and Technology. So also working on magnetic nanoparticle detection and especially for temperature measurement. So afterwards, then I went to La University for 1 year postdoctoral. Then I moved to Braunschweig to do my Humboldt Fellowship, and also afterwards I got my own DFG project. So I have my PhD student to work together with me on temperature imaging with MPI. So in 2021, so I came back to China to got my professorship and start work at Beihang University till now. So, so first I want to shortly describe about or talk about MPI principle, and I think the first speaker from so next MPI this webinar, he has already described a lot about MPI principles. So here I will just shortly describe for MPI. The very important things one is one should need to generate a gradient magnetic field and also with a field-free point. And when nanoparticles they at the field-free point, so if we have a lot of kind of AC magnetic field or sufficiently strong AC magnetic field, you have a lot of spectral signal from the detection coil. But if nanoparticles, which is, for example, not in the field-free point, then you at this kind of the non-field-free point, and then nanoparticles are saturated by this gradient magnetic field, so you don't see any signal from the detection coil. Of course, for FFL, it's the same story. So then, which means that if we scan the 50 points through the field of view, three-dimensional, then we get an image of the spatial distribution of the nanoparticles. So here, the very, very important thing is the underlying physics of these nanoparticles. How can they respond to these AC magnetic field and also this gradient magnetic field? For nanoparticles, normally what we use is superparamagnetic nanoparticles. So when the magnetic field is off, so then the magnetic movement, they are randomly distributed. But when the magnetic field is on, so the magnetic movement of nanoparticles, they try to align to the excitation AC magnetic field, but because they say AC magnetic field, so it's kind of for the field direction which are changed from this direction to the other direction. So then the magnetic movement of nanoparticles, they also rotate to follow the AC magnetic field. So with these, there are two mechanisms. One is near relaxation. So here, like this way, so the nanoparticles, they don't have any kind of physical rotation. So only the magnetic moment they kind of rotate. So we call it near relaxation. Another one is Brownian relaxation. So nanoparticles have physical rotation of it. So then also to make the magnetic moment to align to the AC magnetic field. So these two relaxation mechanisms, they are very, very important to MPI, and also they really or significantly affect the dynamic magnetization of magnetic particles. So because MPI directly measure the magnetization of and also its spectra of magnetic nanoparticles. So here the dynamic magnetization and relaxation of these nanoparticles, they really play significant role in MPI, and also for color MPI. So these two relaxation mechanisms, they are very, very important. So normally for a lot of to describe the dynamic magnetization, there are really a lot of models, and for a lot of like a simplified model, normally what we want to write is to the Dubai model together with the static non-grain equation. So here, this is the basic model. Of course, it's kind of simplified. It has a lot of limitations, but it's still kind of one of the very nice model to describe dynamic magnetization. From this model, you can see, for example, we have the excitation frequency, and also we have the effective relaxation time, and also we have the saturation magnetization, and also this chi, which is the energy of magnetic energy, the ratio of magnetic energy to thermal energy. So for relaxation time, we have Brownian relaxation, so which is significantly depends on the viscosity of the medium, and also near relaxation, which depends on the core size and also temperature. So then we get this kind of effective relaxation. For different nanoparticles, for some nanoparticles, they may dominated by Brownian relaxation. For some nanoparticles, they may be dominated by near relaxation. But also for the other nanoparticles, they are kind of have a coupled Brownian and near relaxations. So for different applications and also for color MPI, one may choose this really different kind of nanoparticles. So this is very important. So from this kind of model, we can know that the dynamic magnetization and also relaxation time, they depends on temperature, viscosity, the status. The status means that when with some nanoparticles, they can physically rotate. But when some nanoparticles, for example, if they are affected nanoparticles with antibody, if they bound to some cells, the antigen, so they kind of immobilize, or so they have the binding status, they can't physically rotate. So it also depends on the signaling changes, their magnetization. And also some nanoparticles, different nanoparticles, they have different saturation magnetization, different relaxations. So it also changes the dynamic magnetization. So there are already a lot of work which did like the MPS, let me say, the spectrum from the spectral side. For example, specimen dark from T. Braunschweig, and also the Jeremy Weaver from the United States, and also PDB group from Wickles. They also did quite a lot of work how temperature changes the harmonics on the spectrum of magnetic nanoparticles, which show that the MPI signal definitely has temperature sensitivity. And also for viscosity dependence, also there are a lot of other group. For example, Turkish group, and also the Japanese group from Takashi Yoshida, and also the PDB group in Berlin, and also with different nanoparticles for magnetic biosensing. People want to realize for the multiplex detection of different antigen or antibodies. So there are several groups that are working on it. Germany, and Japan, and also the United States. For example, the Minneapolis group, also Professor Jianping Wang, they did a lot of work. But all of these show that the nanoparticles magnetization has temperature dependence, viscosity dependence, and different status, and also this kind of nanoparticles, when you have different nanoparticles, the spectra, they are different. So MPI, they measure the spectra for imaging. So in principle, it also allows multi-parametric imaging. So for example, color MPI. So then here I want to kind of summarize what is multicolor MPI. So from my understanding, yeah, so maybe different people, they have different persons, may have different kind of understanding. So it can measure the spatial distribution of magnetic nanoparticle concentration and also probably the local or environment parameters like temperature and viscosity, and also maybe viscoelastic properties, and also the spatial distribution of the status. As I mentioned, when nanoparticles are bounded onto the cells or uptake by the cells, so they either can be physically rotate, and when go inside the cells, they can't be physically rotate. So this is changes probably from Brownian and near relaxation coupled, and then to only near relaxation dominated, and also with different nanoparticles. So color normally use this different color to show different parameters or status or different kind of nanoparticles, and then the intensity use it for or these kind of parameters, the how the value it is. So then I will give kind of a status what currently a lot of group have already done in this field. For example, the first group have done regarding multicolor MPI, I think is the Philip group. They published a paper in 2015. So they have basically two different nanoparticles, but not three, but two. But if we also can treat it like a three nanoparticles, so it's MM4 and also reservist. So MM4, here is in the liquid, and also another one is like dried powders. They have really two different kind of status. If you see this kind of spectra, so the red one is the reservist, and then the green one is the MM4, which is also liquid. To the nonlinearity of these two nanoparticles, they are almost the same, but the powder of MM4 is totally different. But if you look at the phase, you can see that the reservist, which is totally different from the MM4. So which means that because MPI normally would not just use the amplitude, but also to use the real and imaginary part. This is very important, to which demonstrate that the kind of spectra of nanoparticles, these three different nanoparticles, they are almost they are very different. So then they have a kind of a phantom of this one reservoirs, MM4 and MM4 powder in different places. They have only one scan, and then you can make all of this kind of spectra, and then get kind of for color MPI, you get a kind of different kind of, let me say, the forward problems. So the vector V is what we measure the spectral signal, and then you get kind of the system matrix multiply with its concentration, and with 3 you get another kind of reconstruction, inverse problems for reconstructions. So finally they demonstrate this is the kind of black white image, and they kind of overlay, you can see this reservoirs, MM4 and MM4 powder, so you can really realize powder. But here you also can see that with each different color, you have a lot of kind of channel linkage to each other. For example, for this color, you can see that you see, especially this MM4. So you can see in principle, it should be only have MM4, which is liquid, but it's also have some color from the reservoirs, and also the other way around. And here, the very important thing is here the 3 different nanoparticles, they already separated and different spatial position. But especially when these nanoparticles, if you have the same positions, so you will have a lot of other kind of trouble to really can realize quantitative or image of this the concentration of these 3 different nanoparticles. So basically, we need very nice reconstructions algorithms, and we also need very kind of nice nanoparticles to make it the spectra are very, very different. So then you are able to distinguish these 3 different nanoparticles. And also for temperature imaging, this is also from Philip Group. They did it in 2016. So they have they calibrate the system matrix of one nanoparticles which at two different temperature. For example, T1 and T2. One is the lowest one, I don't remember, maybe 20 degree, and the other one is above 40 degree. And there they get kind of use color MPI to get reconstruction problems like this one. And then for this, you can reconstruct the concentration and temperature T1, and the other one is concentrate spatial distribution of the concentration and temperature T2, and then they simplify the model to get the kind of difference of the temperature, which can be calculated by these equations, and then they do reconstructions engines, and you can see like this is without heating, so they are this one, and then with heating you can see totally this kind of different temperatures, the spatial distribution of different temperature. So meanwhile, we also did quite some work regarding temperature, so I will discuss it later on. So for your viscosity imaging, so there are a lot of work about it, so I will just give a couple of them. So for example, from I think from Tobias Klopp from Hamburg Group, they published a paper in 2018 and in New Journal of Physics. So reconstruct problems like this way. So C1 is the kind of concentrations of the lowest viscosity, and C7 is the highest viscosity. So you know, for example, calibrate the system matrix at very low viscosity, maybe just in normal or DI water, and the other one is very high viscosity. So you treat this kind of as a kind of linear curves, and then you do this use color MPI to reconstruct C1 and C7, and then they got all of these kind of step by step, all of these different viscosities, and they use a method to calculate, and they got the spatial distribution of also calculate the value of viscosity. So there are a lot of other statistics data. If you are interested, you can go to this paper. And another one very interesting work, which is done by X-based MPI for viscosity imaging, I think from Emily Group from Turkey. So for X-based MPI, basically you can kind of measure the time domain and signal. And with this time domain signal, you need to do kind of a deconvolution with this kind of time relaxation behavior, and then you can get a spatial distribution of the relaxation time. So this is called tau maps. And there, because tau maps a relaxation time, which also depends on the excitation magnetic field, that we call it's kind of effective relaxation time. So also you calibrate the curve of relaxation, effective relaxation time versus viscosity and different AC magnetic field, and afterwards then you kind of align all it, and then you get kind of a color MPI for viscosity. So this is the viscosity. This is the color MPI, color imaging of a relaxation time, and then you can calculate it to a viscosity. So this is also very nice. They also published several other papers in, for example, physics, in medicine, and biology. So if you're interested, you can go this here. And another one work which is very, very interesting from my point of view is also published by Hamburg Group, especially from medical hospitals. They have kind of animal models with brain bleeding. So you use called multi-contrast or multicolor MPI, which can really distinguish the blood is bleeding or liquid, or it's already become a kind of solid, so coagulation. For bleeding, the magnetic nanoparticles, they can physically rotate, but when it becomes a solid, so it's kind of coagulation of the blood. So then you only have physical, no physical rotation, but only have near relaxation and domination. So here, these are three very important videos which is produced by them, which is very, very interesting. Yeah. So then also another group from PDB, so from Course, they when you incubate nanoparticles together with cells, so cells they eat these nanoparticles. So when nanoparticles in the liquid medium, they can physically rotate, but it goes inside to the cells, it become like immobilized. So they really can distinguish nanoparticles gradually go inside to the cells. So for here, especially here, the intensity of the image of these mobile nanoparticles, they decrease, and here they increase. So this is very interesting. I think also have a lot of biomedical applications. So for my work at afterwards, so I will introduce my work at TU Braunschweig. So we use scanning magnetic particle spectroscopy called SMPS. So we can do mechanical scanning. So there we can measure the spatial distribution of each harmonics. For example, the fundamental, the third, and the fifth, and so we want all of these. So here we made the spatial distribution of the fundamental harmonic and also the third harmonic, and then we get a ratio of the third over the fundamental. So which then that is only dependent on temperature, but independent of on concentration. So we can get all of this temperature imaging with different heating. You can see different places they are heat up. And afterwards, basically, we do concentration imaging and temperature imaging. And basically, in principle, they also can be done like we really call multi-parametric imaging, like concentration, temperature, and viscosity. So we proposed some kind of phenomenological equation from Fokker-Planck equation with Brownian relaxation, like this way: the third over the fundamental, and also the fifth over the third. So they for viscosity, they only depends on the multiplication of frequency and also the Brownian relaxation. So if we change the viscosity or you change the frequency, they are more or less like the same story, like this way. It's eta multiplied with the efficient frequency. And there, this is very important for this multi-parametric imaging. Then with three different dots at the same image, so we can, with different viscosity and also different temperature, so really can get a kind of spatial distributions of viscosity and imaging and also concentration as well. So here from all of this, we can do that the spatial distributions of harmonics, they are very, very important to realize this kind of multicolor MPI. Also, it depends on how we can work. So then because there is kind of mechanical scanner, so it's kind of MPS with mechanical scanning. So then we go to electrical scanning with general MPI. So here we propose called single harmonic based nanoMPI. We only measure the third harmonic, so the spatial distribution of the third harmonic for imaging. So you can get kind of points reflections of the third harmonic like this way. The red one is with the same direction of the excitation magnetic field. So the blue one is the potential direction, and there we do deconvolution of the third harmonic. Then we get the kind of image of the magnetic nanoparticles. So this is a scanner we built, especially TDFX built, and I modified it with according to this idea. So then we made modulation transfer function, and also we did quite a nice image of the research into the EMG. Yeah. So afterwards, because there with this idea, we can just do this concentration imaging with either the real part of the third harmonic or the imaginary part of the third harmonic. So here then we measure the temperature sensitivity of this phase lag of the third harmonic. So this is the calibration curve of the third harmonic phase lag. So then we measure the spatial distribution of the phase. So here is the third harmonic. So we do kind of deconvolution of the real part of the third harmonic and also the imaginary part of the third harmonic. So then we get kind of spatial distribution of temperature. So this is a class, and he used to be my student, worked together with me, who did this quite nice work and published in JMM. So afterwards, then in 2021, I came back to China. So I still continues working on these directions. So one of the most important things I built is a lot of calculation tools with for magnetic nanoparticles, like AC susceptibility, to which really can measure the susceptibility for frequency from 100 Hertz to 1 megahertz, and also the MPS setup from 100 Hertz to 32.6 kilohertz, which can really meet all of this spectral and the dynamic magnetization in curve. The strength of the AC magnetic field can go up to 25 millitesla. So now we have a lot of collaborations with material scientists. So they say this is the nanoparticles, and they characterize with our setup and to evaluate nanoparticles is good or not good for MPI. And this is like the limit of detection test. So we can go to about 2.3 nanogram ion mass. So that ACS is just one direction, extension magnetic field. So then we also build this 2D MPS setup, which want to kind of mimic this magnetic field in MPI. So then with different trajectory, how this spectral changes, and it very, very interesting. But how to understand it and also correlate it to MPI performance is quite somehow difficult. So afterwards, we go to MPI. So for basically the frequency domain or space or F space MPI, you made all of the spectra like for 1D, all of the spectra, and then you kind of made the system matrix. And for X space, you don't do any kind of reconstructions. But here we want to combination of this F and X space. So in X direction, we have this kind of general F space. You made all of the spectra, but to improve the kind of time consuming problems regarding system matrix measurement. So in Y direction, we changed the free point with a coincident magnetic field. So the free point is slightly changes in this direction slowly with 1 Hertz triangle or wave magnetic field. Then we do reconstruction in both directions. The X direction we use normal system matrix. In Y direction probably is kind of deconvolution of the free point. So, but then we still get quite nice spatial resolution, but all the hardware cost is decreased to about a half. So this is the this is the kind of system matrix, and this is the scanner concept, and also then we get the next direction spatial resolution is about 0.3 millimeter in Y direction, because the X direction is the excitation magnetic field, and Y direction do have the drive magnetic field. So then the spatial resolution is about only 0.7 millimeter, which is already quite nice. So the gradient magnetic field in X and Y is about 1.1 Tesla per meter. In the Z direction, so main gradient is about 2.2 Tesla per meter. So then we also think about this is kind of for their case, we need to really do kind of a time domain signal segment. But then if we continuously increase this excitation frequency from 1 Hertz to about 100 Hertz, so also you have kind of sinusoidal wave. So there you have a lot of like this kind of mixing frequency spectrum, which is very similar to the MPS, so mixing frequency MPS, like the Minneapolis group they do, and also some of the German group they do. So for this, you have very interesting kind of trajectory of the free point. It's also kind of a mystery trajectory, but then all of the signals somehow is a little bit different from the traditional mystery trajectory MPI. And with this, then we also do a lot of work regarding this phantom experiment, like two dots in X direction and also in Y directions, and also with a little bit complex dots like this kind of phantom. So you can see quite nicely MPI images of it. So afterwards, now one of very, very important things is for us is really go to how MPI, for example, to go to biomedical applications. So we do kind of ex vivo MPI. So for a 2D MPI, it could take us 1 second, and for 3D, it about 3 or 4 minutes. And this is the animal equipment. So we inject nanoparticles from the tail here, and then you have a tumor here, and this here, the other direction is the heart. We can see the kind of a tumor. And this is the very nice experiment results we have done maybe just 1 week or 2 weeks ago, together with our collaborators. They think this is quite nice nanoparticles, which have very long blood circulation time. So you can see that with time going on, you really see the MPI signal gradually increase. And also we do, for example, we start this animal, and then we do liver and spleen, lung, and kidney, and so on. So now biomedical application of MPI is one of our main focus. Of course, meanwhile, we also are improving our scanners. And meanwhile, also do like color MPI or for temperature imaging, we are continuously working on it. So maybe in the near future, in the following couple of weeks, we'll have another paper submitted. And there here we have 2 two different nanoparticles, perimag and sinomag. We want to distinguish these two nanoparticles. We really mix these two nanoparticles together with different percentage. For example, one is gradually increase in the concentration, another one is decrease in concentration. And then we really can see this color MPI. So from here, it's increase and decrease. And then we did the statistics, how quantitative is quantitative. So we can get a kind of linear curve of these different concentrations. So from here, you can see very nice. It's better than 90%. Of course, this is for phantom experiment. For in vivo experiment, it will be maybe difficult to get this so high quantification, but we will work on it. So basically, for my group, so my goal is to multicolor MPI, especially to want to develop new methods and instruments for disease serologistics. So here the instruments, of course, is MPI scanners, but we have somehow we have a lot of collaborators or contacts with medical doctors and also some scientists from biomedical or biomedical engineering department, and also for some people who develop new drugs. So we want to develop different kind of methods and instruments of MPI for their application. So it's kind of application oriented. As all of you, MPI is a really kind of interdisciplinary research. So it really matters regarding like chemistry, material science. They synthesis nanoparticles for vaccine and also vaccinations. This is very, very important for MPI. So we have collaboration with some scientists in this area, and also physical modeling, like a Dubai model for Planck or non-differential Gilbert equation to understand how these nanoparticles, the magnetic movement, they rotate or they follow the excitation magnetic field, and of course the kind of electrical engineering, electronics and instrumentation, and also reconstruction. We can use traditional like Kashmar's method, and also nowadays very fantastic one is the AI-based reconstruction. So this is also one of the topic we are currently working on, and also biomedical applications, of course. So we have a lot of collaboration, as I mentioned, with medical doctors and so on. So we have, my group now have a lot of facilities on MPI, so we can characterize, if you think that this is nanoparticles, we can characterize these nanoparticles with ACS and MPS. And also we have different kind of trajectory, kind of field sequence MPI scanners, which allows to do animal experiments. And we are also developing or designing another kind of MPI scanner, which combined with magnetic hypersmear. So this is also in plan. And also we have a lot of data for image reconstructions with AI-based reconstructions and also traditional ones. And also very, very important since we have facility to do animal experiments. So for all of this, we are eager and open for all international collaboration on MPI. So if you're interested, to just contact me. And very important since we welcome all applications of PhD student and postdoctors in my group. So then that's all for my talk, and I would like to thank all collaborators around the world. I've done a lot of work in Germany. I have a lot of people from Germany, and also a lot of support from different kind of funding agency, and also DFG, and also Alexander von Humboldt Foundation. So that's all. Thank you for your attention.
Fantastic. Thank you so much, Jing, for your presentation. Let me see. Please, if you have any questions for Jing, we have some time left for this webinar. So anyone with a question, please raise a hand or write your question in the chat box. Anything is okay. So while waiting for anyone to raise a hand for answer a question, I do have a question in the meantime for you, Jing. If you could go back to your slide 24 on your in vivo imaging, it would be easier to talk about it. Thank you. Yeah, that one. I'd love this image. Great work here. And I do have two questions. One is on your scale. Basically, you do have a scale, but the question is it the quantitative? There are colors in there, fantastic, but are they also quantitative?
You mean which figure?
Well, I do see overlay in the images, and there is a heat map, right?
Yeah.
So do you also have numbers to go with those colors in a heat map? So are they quantitative? Are the pixels and the colors in there quantitative as well?
Oh, yeah. I mean, we are still working on it, and we want to afterwards we get the kind of the tumor tissue and do this to measure the iron mass of this tumor tissue and also correlate to it. Currently, from what I understand, it's quite quantitative, but it's always a question how quantitative is quantitative.
Of course, of course, of course. But so as far as I understand, you're working toward a quantification of this heat map as well. So that's not done yet.
Mm.
Is that correct?
Here the heat map is the concentration of the nanoparticles. It's not like temperature or whatever
No, I
else.
understand. That's a concentration. That's something I would, from let's say cell tracking, etc. I've seen one question coming in a chat box. Please, Gabriel, take care of it in a moment. Let's figure out this.
Of
So I
course.
No,
understand it's the
we
concentration.
yeah. So we
This
don't
would be,
have any kind of sorry, yeah,
so you
yeah, please.
don't know what is the concentration on, let's say, light or on yellow and dark red. You don't have that information.
Currently, I mean, I asked my student to do that to better to give, for example, the concentration is how many microgram per milliliter
Yes,
and so on. But because we do this kind of systematics reconstructions, so afterwards is always a kind of relative or value. But we need to just do one kind of how should I put it, a kind of calibration image, and then we can get to all of this, make it quantitative.
exactly. So that would be my question before you're going into, because that's very recent work on your side, before going into
Yeah,
publication, would it be wise to have some kind of lookup table? And that might not be absolutely sensitive or exact, but it would be nice to have through lookup table to have some kind of measure, quantitative measure on the amount of iron in there, because from let's say application perspective, we talk about cell tracking, etc. That would be information people will be really, really keen on having.
Okay, thank you for your suggestion. I will ask
Sure.
my student to work
Gabriel,
on it.
brilliant. We could talk about that later, but Gabriel, please
Yeah,
go.
yeah.
Of course. Thank you for your presentation, Dim. We have a question in the chat from Mrs. Demirel, and her
Yeah.
question is, do you have any targeting agents on the surface of the magnetic nanoparticles in your cancer study, like
No,
tumor imaging?
no. Currently, the nanoparticles all from EPR effect. They go to the tumor. We haven't fractionalized with, for example, antibody or some kind of aptamer. It's not.
So
So we don't
not
have any kind
yet,
of
but that
not
will
yet.
come, I suppose. Are you open for that
I
kind of collaborations?
Yes, sure.
Okay, so then we can say that you also open for STSMs for next year, maybe?
This kind of nanoparticles is it's not a commercial one. It's from our collaborators. So this nanoparticles because of some, I think this is a very very nice one. So we haven't had it for fractionalization. But when I also discussed with him, it would be nice to have also some antibody fractionalized, and he worked on a lot of like nanomedicine, and then he said, okay, this stage probably we will not, but in future we will he will work on it. But as just Leila said, we were very happy or very interested to all of this collaboration like this point. For example, if we have other scientists have worked on this um, synthesis these new magnetic nanoparticles and also do the fractionations with antibody and so on. So we can also do these characterizations and also animal experiment to evaluate how good they are. I mean, for a lot of nanoparticles, when we discuss about how good these kind of magnetic nanoparticles they are, is always a lot of question about it. The first is about how non-linear of this magnetic nanoparticles, so you get a very strong signal from the MPS. But then the problem is whether these nanoparticles, if you inject or they immediately probably they will go to the liver, but they never go to the tumor tissue. To them, this is the problem. To them, for us, I mean, in normal just DI water, they very nice, but maybe they will not go to the tumor tissue. To them, for in vivo experiment, they maybe it's not that good. Yeah, for animal, at least not that good for this tumor detection or application. Maybe for good for other applications. This is also another
The
thing one need to think about it.
Targeted treatments are also very, very
Yeah,
interesting.
yeah.
Well. There's a lot of things to do there, and I suppose the take-home message from this is for Ms. Demirel and everyone else: reach out, and a lot of is possible, even through SDSMs or sending people to China, to Jing, to do measurements and
Welcome.
interpret them. Yeah, I suppose it is. Any more questions on
We have
in the
another
chat?
one. Since that Jim already said that he used some nanoparticles from some of his collaborations, one of the question is, what is the size or the size range of your magnetical nanoparticles that you're using in your studies?
I think currently I can't answer this question because my collaborator is still working on it. But the guess is about the nanoparticles, which is about for single one is about if I remember correctly, he mentioned 15 nanometer. But of course, it's not a kind of single nanoparticles. Afterwards, they have another kind of polymer to
I might be able to provide some clarity on that based on nanoparticles you use, like the perimag and cinemag. That is usually you measure the size on either core or on hydrodynamic, but in terms of core size, we will be talking about very small nanoparticles in terms, let's say 5 to 10 nanometers to 150 nanometers range. But there are different nanoparticles usually, and usually nanoparticles we use in MPs are, let's say, close to monodisperse. So different nanoparticles, there would be like 10-ish, one nanoparticles, and another one like perimag is quite large, like 100-ish something nanometers, and but quite narrow in dispersity, size dispersity as well.
We plan to submit this paper maybe before the end of this year. So when it's published, so we will give all of these details of the particles of the experiments. But otherwise, I can't say too much about it right now.
Brilliant.
We
Thank you.
understand.
Yeah, we still have 15 minutes to go for more questions. And in the meantime, well, we're looking at this image. And again, I love how you're moving forward towards real applications and thinking about a step forward as well. Now we work with mice and rats, etc., with rodents, and we're going to have to start working with, let's say, real clinical applications and patients. And what I see here is the visual camera and superimposed MPI image on it. That will not work in a clinical setting, probably when we start talking about deep tissues, etc. So having PET CT and MRI in mind, what do you see as a good thing moving forwards into real clinical application? Are we talking about something similar, visual information and superimposing MPI, or kind of color image, or are you thinking about some structural information from CT or MRI?
Yeah. So first, very good questions for all of this, and the main point currently for I would like to say that we are working on this. So 2 points. First is of course currently my main work is working on this preclinical MPI scanner. For all in future, so if I have enough space and also enough funding, so I also we will go to like the human brain MPI scanner. So this is also my plan in future, but not right now. The other point is regarding, for example, with PET CT. So I have already built a lot of collaboration with some medical doctors from hospital in one university. So this kind of university affiliated hospital. They have all of the facilities for PET, for CT, and recently we are also discussing about how, of course, the kind of overlay of CT and MPI, very important. CT provides you all of this structure, or MRI, but also whether can we, for example, do a kind of multimodal imaging, which also combine PET image and also MPI image. And for the combination of these two images, what additional important information we can get? So we are discussing about it. So currently we haven't got any kind of good ideas about it. But I only can say that. We're thinking about it and hope that we find some interesting ideas to combine the PET image and so on and MRI, but we will see.
That in your mind, that would be just having two images and fusing those afterwards in software. Is that correct?
Yes.
At this
Well,
point.
of course. Yeah. But of course, the group from, I think um from Folkmar Schulz group, they used to have a kind of hybrid scanner with combining this MRI and MPI, right?
Yeah, there are, to my knowledge, there are more groups. Turkey, one in Turkey as well, where they try to combine low field MRI with MPI as well, which actually goes more of the direction of PET CT, but not because in CT in PET CT hybrid is not a diagnostic CT. It's not high quality, high resolution CT. That would be something we could pursue as well. Just another check. Are there any questions as we move into the
Yes,
last
there
10
is
minutes?
another question in the chat with Mr. Silva. So thank you for your presentation. What is the time resolution of your 2D MPS, and how does it correlate with the used frequencies? And lately, how big is the field of view in 2D?
Okay, so this 2D MPS, because it's really mechanical scanning, so you really have a very large field of view. So first, yeah, so it's really kind of just a mechanical movement. So I don't know, there we have a very big one. So it's, I mean, it's, we can go to something like several tens of centimeter field of view in 2D. The third direction does not work. Yeah. So then the other one, how does it correlate with the used frequency? So here, the frequency, if we talk about the used frequency of the extension magnetic field, so then it does have no kind of, not really kind of correlation with the time resolution. I mean, the how fast you can get 1D image. So they are somehow dependent. So therefore, my case, what I did is I have this trajectory scan. The mechanical scanning like go to line by like this kind of like Cartesian trajectory. So for one line, it takes about 2 or 3 seconds, and then it depends on how many lines you want to go. So then it takes some like I don't know, I don't remember clearly, maybe about 5 minutes. I would like to say in the order of 5 minutes for one 2D image. So there we have their 2D MPS. We really go to different frequencies. We have a kind of relay of the matching capacitor, which go to from some 100 hertz to 10 kilohertz, something like that. To also can characterize the nanoparticles, but also can go to kind of 2D.
How
image
many
of the nanoparticles.
excitation frequencies are we talking about? If you do 5 minutes for one 2D
One
image,
frequency, one frequency.
for one frequency, and then
Yes.
how many excitation frequencies do you do? It depends on the frequency resolution, of course.
Yeah, it depends on frequency, but normally if you just look at one D image, then just need one frequency. It's already enough. But of course, if you want to do, for example, this viscosity imaging or temperature imaging, so then you better to have several different frequencies.
But are we talking, Jing, are we talking about like up to 10 different frequency the excitation frequencies, or are we talking about up to 100? Because there is significant difference. If it's 10, then we're talking about an hour.
No, I haven't done with 100 different frequencies. I think it depends. Probably it's not necessary to do 100 frequency MPS. Normally, with even with SES, I would do something like 50 different frequencies to really sweep the frequency. But roughly in the order of 10, so maybe maximum 20 for MPS. Because to have a relay of these capacitors, it's a lot of work. Yeah, it's not difficult, but a lot of work.
Yes. Well, I think it's always when we talk about spectroscopy, and this would have a big resemblance to magnetic resonance spectroscopy as well, but optical spectroscopy as well. It's always a trade-off between time, resolution, field of view, and number of frequencies you use. More frequencies you use, better quality of information you will get out of it, but then that will run into timing, etc. So I think the first trick is to identify which frequencies are relevant for a specific application and decrease number of frequencies, so you will increase the resolution of field of view, etc., within a reasonable time. But it remains a trade-off as always, I suppose.
Yeah, so it's always, I mean, I think I mentioned it's always the question is from application side or from calculation side, whether we need so many frequencies. Of course, if you say some applications, okay, we definitely need 100 frequencies. With 100 frequencies, we can understand much better than 20 frequencies. Then of course we will do. It's as I mentioned, it's not a problem of difficulties, but it's really just a problem of time. You need to build these relays of these capacitors. So currently we didn't see a lot of we don't see a lot of kind of how should I say a lot of
one last question from my side. How many excitation frequencies do you have functional now in your system?
Okay, 1, 2, 3.
Okay, just up to
But
3.
With 3 different scanners. But in near future, I asked one of my students to work on it at least 2 or 3. With the same scanner, which can run to MPI. I mean, not MPS, 2D MPS, but really MPI, 3D MPI scanner ran to like 2 or 3 different frequencies. Because
can I keep requesting? Can you have them tunable? Let's say at the same time you could do 2 frequencies or 3 frequencies consecutive, but then you should be able to tune them into interesting frequencies for a specific let's say application. If that would be possible, even in preclinical setting, that would be brilliant, at least from my knowledge.
So you mean excite, for example, 5 kilohertz and 10 kilohertz at the same time, or?
No, you could do that sequentially, but then let's say for one applications, it would be interesting to have 5 and 7, and for another application, maybe it's a different nanoparticles, that would be 4 and 6. That is interesting. So if you could have 2 sequentially excitations, but then have some freedom in there to tune them a little bit as well.
In principle, yes. Yeah.
Oh, that will be brilliant.
This is not, yeah.
We have a few more. Yeah, go ahead.
But it's a lot of work. Yeah. So normally, it's really different application. I think for a lot of applications, one of the frequency we want to choose, for example, at rather a low frequency, which is, for example, about 5 kilohertz, and then the other one is a little bit high frequency, maybe 25 kilohertz. I think once Tilo published a paper about it, when you use 10 kilohertz and 25 kilohertz, two different frequencies, you already get a lot of kind of different information, which is already very nice. It does not make sense. For example, one 10 kilohertz, another one is 25, and then you change it to like 8 kilohertz or 22. Then in this case, probably it does not make too much difference.
Yeah, thank you for your answers. Gabriel, do you have anything else in
Yes, we
Luciana's
have an
hands?
extra question from Rui Silva. And what is the, in terms of expected depth of the 2D image? You mentioned going to be 100 centimeters, but what is the lowest field of view and pixel size? And then Rui said that he's sorry for if the questions are out of the scope, and if he can contact you by email to have to have these answers, if it's possible to reach you by email.
Yeah, sure. Yeah. So first, yeah, it's possible to contact me via email, and also I can answer these two questions. So we haven't done the depths of this 2D image. I think currently for that what we presented, the distance between the detector at the nanoparticles, which is about, I don't remember, maybe 5 millimeters. So it's very close to the phantom, and there. So what is the lowest field of view and pixel size? The pixel size, I think there we go to, I don't remember clearly, but it's on the paper. I think it's maybe 0.5 millimeter. I think in this kind of range. It also depends on how should I put it, how fast they want to do this scanning. And also the other point is regarding lowest field view. I mean, when you have a large kind of mechanical scanning, and then you can set, it's just a scan for 1 centimeter, or it can scan for 2 centimeter. Yeah, so it's your choice.
Okay. Thank you, Jim. I think I have another question here from Patrick. So thanks for your presentation, and he has a short question comment about the multi-frequency excitation. What about the non-resonant systems? Which amplifiers, I didn't understand, which is strong enough you can also reach with a high frequencies and high fields? Good question, Patrick.
Okay. Yes. Yeah, good
Tricky
question.
one.
But yeah, in principle, yes, we can use non-resonance systems with quite nice amplifiers and so on. But there, especially for MPI, you will get a lot of problems of nonlinearity. Of course, if you have, for example, the dry field coil with a very low impedance, then you can go to, I don't know, maybe 20 kilohertz, but then you don't need any kind of how should I put it, any kind of resonance system. But this is from the dry field coil side. But then for the detection side, receive side, you still need a kind of how should I put it, a large filter to fit up the fundamental frequency. Of course, the Feng shows his group have done some kind of work with very nice mechanical tuning of this receive coil. There you get a very limited amount of this feed through from the dry coil. There they can do multi-frequency. I don't remember, maybe go up to 5 kilohertz. They published one paper about it, but afterwards I don't know how exactly they can go. Regarding these which are strong enough, you can reach high frequency and high fields. It depends on the power amplifier, and also it depends on the kind of nonlinearity and so on. For MPS, it's rather easy. For MPI, it's again much more complicated and difficult. Yeah, I think Patrick, you know all of these kind of tricky things.
Well, thank you.
Thank you.
In case there are no, Gabriel, are we having anything else in the feed?
Yes, I just want to remember for the participants of this today's webinar that in 4 weeks we're going to have another one with Dr. Leila being the presenter in this time. So uh, just a reminding that you can also use the same link that you enter for the presentation for Jing, and then we can see each other in 4 weeks.
Brilliant. Well, thank you all for participation and the questions. And the recording will be accessible. I'm not sure in what period of time, but it will be at least in a few weeks will be accessible through the nexMPI.eu website. Thank you all. Take care and see you in 4 weeks.